welcome to Reptile Fight Club. My name is Justin Julander, if you didn't know. And with me here is Rob Stone. Say hello to the people, Rob.
Wave to the people, blow them kisses. Indeed, as always.
And we're joined by my good friend, our good friend, Ben Morrill. How's it going?
Good, very happy to be here.
Welcome back, Dr. Ben Morrill. Yeah. For those that may not know, Ben and I were business partners back in the day. We worked together with Australian Addiction Reptiles, and then Ben moved off after he finished his PhD at Utah State, where I also did my PhD work, and moved out to Virginia. So he's been out there doing some science, and you're working with the Rare Genetics Inc. That's kind of your baby now, your company, or at least one of your ventures. You've got kind of lots of pots and kind of keeping all the balls in the air, or juggling on a lot of things. So, I don't know, why don't you tell us about kind of what you're doing these days?
Yeah. So since I moved to Virginia, I did work for a company, a pharmaceutical company. I was doing molecular biology, genetics kind of work, and also some embryo work. I was helping to do somatic cell nuclear transfer, so cloning for some pigs. So we'd do edits, and then we'd clone the pigs, and those pigs then had edits that made their immune system more compatible with the human immune system. And so I did that for 10, 11 years, something like that. And so the last 2.5 or so, I quit that job and have just been doing reptile stuff. And the other thing that happened in 2011, the carpet python book came out, and Justin and Nick took off to Australia and whoever, who knows where else, Europe or somewhere. So when Tinley came around, I was the only one that was around that was a part of that. I wrote one of the chapters in that first one. And so I went to Tinley and sold books, and Sean Christian, who was on the Morelli Pythons radio, or the Morelli Pythons radio, the Morelli Pythons forum forever ago. That's where I started talking with him. And I think that might've been where I first started talking to you because I was looking for rodents. Mm-hmm. That's right. And, but yeah, with Justin, I met, or with Sean, I met him in that forum. So 2011 was the first time I met Sean, and he said, "Hey, I keep birds too. We can pull a feather and send it in, and they can tell us what the sex is. Why can't we do that with reptiles?" I was like, "Oh, because nobody's done the work." He's like, "Well, why don't we do the work?" And so him and I had kicked back and forth things since from 2011 to 2017, just kind of looking what had been getting published and stuff like that. And 2017, we finally had one test available. It was a sex determination test for colubrids that worked pretty well across all the colubrids we've tested, except for tricolored hogs. And then it kind of seems like house snakes we have some troubles with, but most of the other colubrids, it seems to work well.
Yeah.
And then in 2022, kind of early to mid 2022, we started playing with morphs and ball pythons. There were a couple of publications, a group in Canada and a group at Eastern Michigan University. Both did some publications. The first one was the Canadian group. They found the piebald mutation for ball pythons.
Oh, cool.
And since then, they also went in with a group from Georgia and did CRISPR edits in anoles and made piebald anoles.
Yeah.
So pretty cool.
That is cool.
And they also, someone else with that Georgia group did albino anole also. But yeah, and then the Eastern Michigan group, they had a publication come out pretty early on albino and toffee / candy and ultramel and lavender albino. They found all of those. They kind of went and searched specifically in those albino kinds of genes to find those. And so, yeah, from then it's been sequencing costs have come down, and access to bioinformatics has become, it's a little easier to get, off the bottom of people's queue that know what they're doing. And I've been learning some of it too. And so that made it so it was cheap enough and accessible enough where we could start finding some of these things ourselves. So it's cool. What I've been obsessed with for the last 2.5 years, almost 3 years.
Nice. How many tests are you up to?
I think we're either at 60 or a little over 60 now.
Oh, wow. Yeah. That's awesome.
Yeah.
Very cool.
Well,
Is it mostly like mutation, genes to mutant genes and sexing?
Yeah. The only thing we've got so far is colubrid and ball python sex determination, and everything else is all color and pattern mutations in ball pythons. Yeah. Well, actually, that's a lie. We do have, I think, 5 corn snake morph tests now too.
Okay. Yeah. Cool. Yeah, I was. You kind of were the my, I guess, teacher explaining why this stuff is difficult, and how to all the things that we didn't really know about sex determination in snakes, and how it wasn't, some things were wrong in the literature. And so that changed everything, just a lot more difficult than I thought it should be, think, oh, male, female, that should be an easy thing. I guess it's kind of easy in humans, or, you just look for that Y chromosome or whatever.
It's really interesting in snakes that it's so different, because in like all of colubrids, that those sex chromosomes are fairly well conserved, even across, thousands of different species, a couple thousand species or whatever it is. Those sex chromosomes, first of all, they're really different. That's ZW in colubrids, and the Z and the W are much different in size, just like human X and Y. Right. But in pythons and boas, it's XY, and the X and the Y are very indistinguishable. Even if you do chromosome spreads and stainings and all that stuff, you can't tell the two apart.
That's crazy. So
it was very difficult and expensive to get the ball python test. But now that we have that, and we're, I think, finally circling back around to do some more sex determination and morph tests in other species, I hope our next sex determination test development will be faster, but we'll see.
Yeah. And that, it seems like the obvious one would be green tree pythons. I know you've had that in mind for a while and working on it. Is it going to be similar to the ball pythons, or are there differences between, different python taxa?
So from what I haven't seen the data myself, but in talking with Dr. Warren Booth, it sounds like out of all the python genera, the only one that has weird sex chromosomes, like pythons in general have XY, and they're fairly similar. Boas also have XY, but it was an independently derived XY, so really? It's actually different. Yeah.
Okay.
We kind of lump pythons and boas together in our heads, but especially their sex chromosomes, they're very different.
Okay.
But yeah, unfortunately, there's one genus in pythons that has whacked out sex chromosomes, and it's Morellia. So, so that's why it's such a pain in the butt. And it's not just us. Like Warren's worked on it. Other people have worked on it, and it's. I had tests that would work for, I'd try a dozen, and then I'd try 20 or 30, and then I'd try like 50 or 60. And every time the best I could get to is that, 20, 30. When I get to 50 or 60, it wouldn't work across all of the samples. So it just wasn't it wasn't a test that worked across all animals. And it could be that it's a species or locality thing, but it's so hard to know for sure where samples are coming from,
Yeah, that's interesting, because aren't ball pythons kind of the more basal of the group? Is that still the thought on the pythons? Is that ball pythons are kind of basal, and like the aspidites are the more derived? And
I believe so. Yeah,
kind of that trend through to Australia are the more recently derived species. But yeah, that's interesting. You'd think they'd all kind of keep that, as there. But
and the fact you can make a carpet python, ball python hybrid. You can do the Woma ball python hybrid.
Yeah,
but
you'd think
apparently there's something different enough. And so we're definitely working on, and that's the exact test that Sean and I were talking about. Since we are Morelli people, we wanted to make a green tree python test. So that's by far the number one test we spent the most time and the most money on that we still don't have to this day.
Shoot.
For a while there, probably for a year or two, I had a test that did work. And this is when I was still working for the pharmaceutical company. And so I could just kind of do it, not really charge for my time, but just kind of charge for materials. And,
right.
So I did work out where if they sent me the sire, the paternal grandsire, or the male offspring from that sire, and then they sent me all the offspring that were the unknowns, then I could use a marker on the X and Y. I could use a marker and I could see in a known male offspring or the paternal grandsire what marker was passing to males in that line, and then I could tell which of the babies were males from that, and then the others would be females. But it's like now that I'm doing this commercially, I definitely can't do it for less than $100. It might be more like $200, right, because of the analysis time and stuff.
But with the amount that they're selling captive bred green tree pythons for, that might be worthwhile to get those, get that determined early.
Not when they get the answer they don't want, Justin.
That's true. Yeah, I guess if it's part of your
of it.
Yeah, that's true. Well,
but I'm hopeful. I'm hopeful we can figure it out, but we'll see. Yeah. It's not an easy one, that's for sure.
Right. Oh, well, yeah, good luck, and hopefully you'll have some kind of breakthrough that helps you, make it a easy, simple test. I'm sure that's the goal, but
yes.
Yeah. Well, now do you have employees? Are you doing pretty much all the sequencing and all that yourself, or do you have other people working as well?
Yeah, so we've set it up. We have a pretty good sized lab in Texas. So I'm pretty much just research and development.
Oh, cool.
And nice. Then Kayla that goes on ours. So we have a weekly YouTube show.
Okay, yeah.
On Tuesday nights. So Kayla's the person that helps me get that recorded, and she knows how to do all that editing and get it all up. So but cool. She definitely helps me like sorting sheds and with some of the stuff. But yeah, if it's getting a new test working, that's me. As soon as it's working, then I send the stuff to Texas. And so if you're paying for a sample to get tested, then you'll send that to Texas. And so yeah, that's the flow for now.
That's cool. And I'm sure you've got they running plenty of tests and doing plenty of samples these days. And it seems like everybody knows who you are, and you guys have gotten the word out pretty well. And
so far so good.
Yeah.
It's a lot different than working I had an extremely safe job before working for a pharmaceutical company. I saw people do really dumb things and not get fired, but I was obsessed with it. I was always thinking about how I could use the stuff we were doing for reptiles. I didn't want to do it for other stuff.
Right. Yeah. That makes it tricky for sure.
But so far so good. Hopefully it'll stay that way.
Yeah. Nice.
That's great. Yeah, honestly, so it kind of hit into the conceit of the idea of the show in terms of talking through the sex chromosomes, the difference between colubrids, boids, pythons. That really hit on it there to me. So the idea is that all of this is much more complicated, or it can be much more complicated than we as average hobbyists like to think, right? Or often how we've taken it historically, and the fact that, there's sort of an unwillingness to believe that, science changes based on the presentation of new information, right? And, sometimes it's always taken as ill intent. It's never, actually the information changed, these sorts of things.
Pre-2017, we thought all snakes were ZW. Everybody would have told you. And even Dr. Warren Booth, I remember I was on I think it was a chat thing or, a forum or something. He's like, there's no way pythons are XY, but then he was the one on the paper when he said they're XY. So it happens.
Yeah, exactly right. And that's not a bad thing. Embracing that reality is awesome.
Yep. Yeah. And I mean, I guess you how do you know, how do you tell between XY and ZW? What are there common markers that are, is it the same XY that we have kind of idea?
It's kind of funny the way those names came about in genetics. So they wanted to be able to make the chromosome name tell you what the heterogametic sex is. So in humans, our heterogametic sex is the male. We have the X and the Y.
Yeah.
And so any animals where the heterogametic sex is a male, they call those X and Y.
Okay.
And any species that the heterogametic sex is a female, they call them ZW. So there can be some things, like if you look at chicken sex chromosomes versus like a knoll, there's a lot of homologous sequence that's on the sex chromosomes between those two.
Sure.
But like if you go from like, an knoll to a human, then there may not be very much. I've never actually looked, but there might not be much similar at all. So just because you call it X and Y doesn't mean the same genes are there.
Right.
It's just telling you what the heterogametic sex is.
See, I miss these conversations. I always learn something new from you every time I talk to you. So yeah, it's hard having you on the other side of the country. But yeah, that's crazy that you just think, oh, XY, but that makes a lot of sense that it's XY because the male is the heterogametic sex. That's okay. That clarifies it. And I mean, they could have completely different structures, right? Like the X and Y of a snake versus the X and Y of a human. Obviously, the X and Y of a human is easy to tell apart, whereas the python's not so much. Yeah. Okay.
Oh yeah. And then there's some species that have like 3 X's and 2 Y's, and there's all kinds of really funky stuff. And then if you look at plants, you have tetraploids and like even more complicated stuff. So when you're talking about just 2 sex chromosomes and it's a genetic thing, sex is a genetic trait.
Yeah.
That's about as simple as it can get. But then you get like bearded dragons where if you cook their eggs at a certain temperature, you can actually have sex reversals. So they're genetically should be one sex, but you cook them the wrong, I can't remember if it's high or low. I think it's high.
Sure. Yeah.
You cook them at a high temperature, it'll actually change the sex. They'll genetically be one sex, but they'll actually be a different one. So yeah, there's lots of weird stuff.
Yeah.
Yeah, so I want to put a pin in that one. I have that one much further down the road, and I know we've talked about this going back 8 or 9 years in terms of, I know that hasn't been seen in snakes, but I have my lived experience suggests that maybe there's something there. And I know we had talked, sent sheds back and forth and want to talk through what it would look like to try and illustrate if we're seeing sort of a similar thing there. But yeah, I mean, in general, right, all of this fits into the box. The conceit of the show is just that our human impulse is to simplify and try and understand things in the most simplistic terms. And even when we're faced with sort of almost incomprehensible levels of complexity, right? Then our answer is, oh no, someone's, leading us astray, or they're just not being, they're being dishonest or whatever it is, as opposed to, acknowledging the awesome complexity of the natural world, right? So that's basically the idea. So I have a whole host of specific kind of questions as jumping off points and things, on that. The first one that jumps to mind, and really what the prompt for me is, what's going on with banana ball pythons? Do you have a test for that? Do you see that? Do you know where it's at? All those things. I guess we'll start there.
Yeah, so we do have a very good knowledge of like the general area where it is. Since it's so easy to see, it hasn't been a high priority to develop that test. Yep. We still plan to. And the number one question that people have is, are coral glow and banana like genetically identical, or are they actually different? And we've been surprised sometimes with Phantom and Mystic. They actually ended up being the same. With Lesser and Butter, they ended up being different. So, it's kind of hard to know for sure until we actually sequence and know exactly what's causing the mutation. So yeah, with banana, the interesting thing with that is it's a really good example for people that are into ball pythons and know the percentages breeders get. So if you have a male banana that has a male offspring that's a banana, that banana mutation is on the Y chromosome, and it's close to the sex determining region, but it's not in the same gene. And so crossing over can occur in between there, but it only occurs about 5% of the time. So if you have a male from a male banana, so it's on that Y chromosome, then that breeder banana male, any bananas you get, it's a 95% chance they're male. and any non-bananas you get, it's a 95% chance they're female. But every once in a while, you'll have that crossover event happen, and you'll have what ball python breeders call a male maker male banana produce a female. Like, you know, maybe one in every
same vein,
couple
right?
clutches.
It all started with females, and
Yep.
or all these were producing females, which, again, take yourself back, 15 to 20 years in the time machine. And now Kevin's lying, Brock's lying, they're full of it. This is a big marketing scam. This is the idea. Right.
Oh yeah.
So those original females, right? That was pre the crossover, right? And so they're all producing females. Is that sort of the situation?
Yeah. So the same thing. So if you have a male from a female, then that came because the female has two X's. So that banana mutation has to be on an X chromosome. Now, as a male, when it produces offspring, it's going to only make female bananas unless that crossover event happens, because every time it sends the X, it's sending banana. Right. And all of those are going to be daughters. But every once in a while, one out of, 20 or whatever, you'll get that switch. So.
And so you've done the math, the statistics, and shown that it's kind of that 5%.
There's actually a paper someone else did. Chris, I want to say, was it Mallory? Something similar to that. But there was, I think he got data from Kevin at Nerd, if I remember right. Okay. But there is a scientific publication. Like if you search for banana ball python and sex linkage, that it'll come up and you can read it. But yeah, they have some nice figures where they show, like, the male and the female and the pairing, which one's the banana, what the offspring were, and they show a few generations, and they show like the percentages and all that kind of stuff. So, and then we did a video recently where we broke down all of the percentages. So like if you breed a banana to a normal, if you breed a super banana to a normal, if you breed a banana to a banana, if you breed a super banana to, but like all the various combinations you could do. Yeah. And then broke down like on the side, and there's a publicly clickable link you can go to, and you can just see all of the different percentages and that kind of stuff. So,
oh, that's cool. So I didn't realize I'm so far behind. I didn't realize there was a super banana. So if you have a super banana male, then are you getting all bananas offspring and they're both sexes of banana?
Yeah. So if you have a super banana male to a normal, then they're all going to be bananas. But if you have a super banana and breed it to a banana, then it depends on whether that super banana, let's see, how does it go? We did it in that thing, so I have to remember. But so the super bananas will have different sex ratios depending on where your banana gene is in that female. So I guess in that case, so if your super banana is a male and your banana is a female, then your sex ratios are going to be different than if your super banana is the female and the banana is the male. And then if the banana is the male, it depends on whether that banana mutation is on the X or the Y. That's going to give you different super banana percentages.
Okay. Wow.
Yeah. So it does get a lot. Crazy. I was surprised. I thought, that somebody else laid out, Kenny from Bomb City Ball Pythons. He laid out all the stuff, and I thought, okay, I'll jump on for a half an hour. It took me like 4 hours to do all of the different percentages and things. It was a lot more complicated than I thought it was going to be.
Right. So is there a visual difference between the super and the regular?
Yeah.
Okay.
Yeah. They're even lighter and more washed out. The bananas, to me, are more pretty. There's more color and contrast there. Right. But obviously as a breeder, it's nice to know that every baby's going to have banana from it. So.
Yeah, that's cool. Yeah, that's one of the more striking morphs that I actually got one in trade at a show, and she's like a proven female. I'd like to breed her to something down the road, but just they're really pretty snakes. She's like a banana inchy something other, like 3 or 4 genes in there, something. But
and she's good
looking snake.
Or with banana. I mean,
yeah. I've always liked the inchy gene too, so that was kind of. I guess the motivation for that. I was like, I'm not doing ball pythons, but I'll trade for a ball python.
And then that's the other fun part. Like, no matter what your pairing is, if the female is a banana, she's always going to do 50-50 males and females because she has 2 X's. So those X's can go to a male or to a female. So
yeah.
So yeah, if you don't have any knowledge of linkage and crossing over, it just seems like this really weird mess.
Right? Magic and lies. Yeah.
Right.
Magic and or lies. Yeah. No, that's fabulous. The well, and it's also unusual or pretty atypical to have a not a albino, type phenotype that isn't a simple recessive, right? So that was the other part that was so strange about that.
Yeah.
As a gene, and sort of all added to the complexity and mystery of the whole thing, right?
Yeah. And there's a type of albino in ball pythons. They call it rainbow. And the people that have rainbow are really happy and excited about it. But that's kind of the joke is, you're just going the harder way to just make a banana.
Wow.
That's funny.
So it's a recessive.
Yeah.
Yeah. Gotcha. That's crazy.
In the same vein, I know there's been discussion historically around combinations of ostensibly simple recessive traits that are inherited in a simple recessive manner that are more difficult to make than that average sort of Punnett square would suggest, right? So I think it was T-positive albino and ivory blood pythons making those combinations where in theory, right, and really the ivory, it's a matrix, so there's a visual presentation on those heterozygous animals and whatever, but just in terms of the odds of getting that double homozygous recessive, that is where you'd think it'd be 1 in 16, it was taking more like 40, 50, 60. In leopard geckos, there was something, I think it was one of the albinos in with a pattern mutation, something like that. I don't remember the specifics. This takes us back, 8 or 10 years ago, and it was on a Blog Talk hosted show that you can no longer find since Blog Talk went under. But I think he was talking about it there, and where you would anticipate it as a 1 in 16, and again, only 2 eggs at a time or whatever, but he had lots of animals. I think he said it took him like it took 130 to get what should be a 1 in 16. And the thought process is right there that those are probably 2 genes that are very close together. And so it requires a crossover event to split them up to actually have them be inherited independently. Right.
Yeah, exactly. Yeah. That's why I love that banana thing, because at least some people have some familiarity with that. But yeah, we definitely see that with some other mutations. We recently did a video about I think it was monsoon and sunset in ball pythons, and from the sequencing we can see they're on the same chromosome and they're relatively close, and there's only a few breedings. They're both kind of higher dollar animals, and so there's not a lot of breeding trying to make monsoon sunsets, but the 3 clutches that were there showed that there's only, I think it was out of like 26 offspring, there's only 2 that had crossover events out of those 26. So it seems like that's what's going on there. And in that case, we know why, because we know that those are on the same chromosome and relatively close. But yeah, what you're describing, Rob, is perfect. Like, I was on a show last week with hognoses, and a guy was talking about, I think it was lavender, and I can't remember what the other, maybe it might have been lavender and hypo, but anyway, yeah, it was the same. He's like, yeah, if I breed het to het, I like never get it, but if I do finally get like homozygous and it's het, then all of a sudden I can make them. I was like, yeah, that's linkage.
Yep. Yeah, exactly. And saying it right in the sense of, this is relative, to some extent it's known now within the community, but 10 years ago it was like witchcraft, again in the same vein of like, no, this isn't a real thing, they're misleading, misrepresenting what's happening. And it's just kind of, that's what makes it so fabulous to me, the work you're doing and just sort of promoting, hey, there's reasons behind this stuff.
Yeah.
Have you found the hidden gene?
I do have a hidden gene WOMA test.
Well,
I always thought that was an interesting name.
Yeah. What did it turn out to be? Is it just another mutation?
So that complex, that spider and spot nose and WOMA and hidden gene WOMA are 2 different mutations. Oh, okay. But there's at least 11, maybe 12 mutations all in that same. They're all close enough to each other, pretty much all in just one gene, but they're all close enough to each other that they all pass just like their, what like ball python breeders say acts like a super or whatever. So yeah, hidden gene woma and woma are in that same complex with spider and spot nose. And some of the others that are interesting is Wookiee, chocolate, bongo, champagne. There's, like I said, there's about a dozen of them. So it's pretty cool. We found them all right there close to each other. The group up in Canada worked on that too and found them.
Wow.
Yeah, it's a really interesting gene that has so many different, and like some of the supers are lethal, homozygous, some of them aren't, and some of them you get some neurological problems with the heterozygous individuals. Some you'll only get it in the homozygous individuals, and then others you can get the homozygous ones and they're fine. And then if you breed a blackhead to a spider, which are both in that same complex, you get a very normal looking animal that has no neural problems at all. It is a spider, but it's also a blackhead, and somehow it would be really cool. Other out. Yeah, it would be really cool to figure that out, because both phenotypically and neurologically it fixes the problem. So
that's interesting. There's just kind of no end to all the different morphs, and like I'm on World of Ball Pythons right now, just like I've never heard of half of these things, or even close to that. I mean, it must just be like a never-ending supply of fun things to look at.
Oh yeah.
Yeah, that's cool.
Sort of in that same vein, is it correct that there are multiple alleles that present the expression of the, T negative albino?
In ball pythons, or
yeah,
yeah, specifically
in mice there definitely are, right? At mice and yeah. So in ball pythons, when we run an albino test, we have to run two different tests. One is a pretty simple, single base pair change. The other one's more complicated. It's the same thing with ultramel. There's one change that's just a base change. There's another change that's like an insertion or a deletion. I think that one's a deletion, and then lavender albino is an insertion. But yeah, we've found many now that we have to run multiple tests. Yellow belly, there's a -1 yellow belly and a -17 yellow belly, and they look the same in combos, and the ivories look the same. But I think I still have not ever gotten a homozygous -17. -17 is a lot more rare than the -1. Most people, their yellow bellies are -1 yellow bellies, but yeah, so there's definitely a lot more complication there. And several of the tests that we do, we actually have to run two tests to give you your one answer.
Wow.
This is great. Can you talk about allelic traits? I know we've been hitting into that, but can you talk about that a little bit, just in general?
Yeah. So when we're talking about allelic traits, we're talking about, so I guess kind of like a standard definition of what an allele or what two different alleles are. If you're looking at a specific spot, a lot of the time you're talking about at least a part of it. Most of the time you're talking about just a small part of a gene, a few base pairs that you're focused on. But like if you talk about like BRCA1, BRCA2 with breast cancer, they'll talk about different alleles and their propensity to cause breast cancer, stuff like that. So there can be, and even multiple genes that are close by. It can be a lot more complicated, but the kind of way to think about an allele is just if there's two different alleles in one spot, it just means there's some base change or some kind of sequence change that makes those two sequences different. And so it's the same location, the same chromosome, same location, but the sequence is a little different. And so like in ball pythons, if you're thinking about Mojave, Lesser, Butter, Russo, like all of those mutations are all in the same gene. So you have mostly all the same sequence, but in a Lesser, one of those base pairs will be changed. And in a Butter, it'll be a different one that's changed. In a Russo, it'll be a different one that's changed. And so you have these different alleles that all have just like single bases that are different from the others. And all the others, it's normal for everything except for where that one base changes or this little deletion, or like I said, with lavender albino, where there's, an insertion of a different chunk of sequence, but then that interrupts that gene so it can't work properly. So that's really what you're talking about with alleles, is it's different copies of that same area, whether it's a small part of the gene or the whole gene, whatever you're comparing. And it's just, there's some sequence difference between the different alleles.
Wow.
That's great. And this is before we add in the complexities of sort of, there are other genes that present different traits in terms of an animal's phenotype, right? So like just in terms of variance and things, because that was a big part of the discussion was, are they actually different, or is it other traits within the animal that are causing it to display slightly differently? Is it just, the sort of the founder, the average phenotype of the founder stock or whatever with the same mutation causing this different presentation? So it's great that you have actual, you have the answer now. Again, if only we had this 18 to 20 years ago. You're right, you're wrong. No.
Yeah, it would change a lot of breeding plans, especially with the banana stuff. Sounds like you were there for some of that early drama, and that was like $50,000 people were spending, and they're not letting people know all they can make are females. That's why they're selling females, because they can't make a male. And then all of a sudden somebody does make a male, and then they can only make males from that one. They're like, what? And then they're trying to decide whether they're going to tell everyone else, because now they're still like 20 or 25,000 a piece. And those ethical dilemmas that went with that, that had to have been such a wild ride.
Yeah. Well, and not knowing, you can't fault them because they didn't know. Like, you can say, well, most of the ones I'm producing but you can't say conclusively like, oh, they're just making female bananas.
Is that what you would assume?
Because, if you've only produced 8, Yeah,
right? Yeah,
exactly.
And then it's sort of a sample size question. And I do think, I guess this is a open-ended question. I don't know of a different example. Do you know of a different heritable trait in snakes that operates in the same way as the banana? Because at that time, right, this would be kind of the first instance we're seeing something like that. I guess there was a suggestion, right, that maybe these big calico, ontogenic appearance calico reticulated pythons were invariably big females. And so that was kind of a floating around idea. Is cool. This something? But seemingly they were never really, certainly not consistently reproduced. It was these big wild caught animals, but they were invariably these large females. And it was like, oh, maybe there's something to this. But are you aware of any other traits that operate in the same way?
I'm not. And the other fun part to that is like in humans, our X and Y, like if you have a sex-linked trait in humans, we'll talk about, like color blindness or something like that. There's not really any crossing over happening. Like that's always going to stay on the X chromosome. And so that was part of what was so difficult to figure out with banana and the ball pythons, is because there's still crossing over going on. And so that recombination is what makes it so you can get, that 5% different than, that like in humans. I don't know. I have never read anything where that color blindness thing had, spread over to the wild. There's probably not even homologous sequence there to switch with. Right. But that's the other crazy thing is like, as any genetics you go into, you think about sex chromosomes not crossing over at all because they're different. Because most of the species that we're doing experiments on and stuff have different sex chromosomes, and so that took a lot for my brain to figure out. Because I was thinking of it as a sex-linked trait, with very different sex chromosomes, and what was going on just didn't make sense because you can have a trait on one of the X's and then not see it. And that's why there's not very many women that have color blindness, because they have another X there. And so they're completely normal. So I thought for a little while with banana, maybe some of those females are actually bananas. You could breed them and make bananas from them, but you just can't see it. But what I didn't know then was there's still crossing over going on. And that's what made it, that's such a weird thing that so different from normal, mammalian genetics.
Yeah. Do you have an approximation of like the homology between the X and Y in a python versus like X and Y in a human? Because obviously, the humans are very different, and so this idea of crossing, and maybe explain kind of the basics of crossover just for our listeners that may not know what crossover is.
Yeah. So that crossing over happens if there's, and I'm talking with my hands, but this is going to be audio only. Huh? The crossing over, you have a chunk of DNA on two different chromosomes. So it can be chromosome 1 or chromosome 7 or whatever. But that the majority of the sequence in those two chromosomes is similar. but one you got from your mom, one you got from your dad. There's going to be some slight differences, and some of them will matter. They'll make a difference in a phenotype, but most of them aren't going to. But
and
what
that's
happens
homology,
is,
right? The
yes.
similarity between,
the vast majority of the sequence is going to be the same. Like if you compare us to a chimpanzee, a lot of the genes are going to be like 97% identical. Just those few changes. And then in some of the other sequence where it affects gene expression, there's some differences, but yeah. So chunks of DNA that if there's two different chromosomes and they have a bunch of DNA that's almost identical, there may be a few changes, but when those two pieces of DNA touch, come into contact with each other, which they're floating around in that cell, they can easily come into contact. And what can happen is those two pieces can actually cross, like literally kind of cross and actually switch. There'll be a double strand break on each, they'll switch. And so that helps to increase genetic variation. So if there was no crossing over, then whatever was linked, like with banana ball pythons, we would never have that 5% that changed because there would never be a crossover event. And so that's part of when you're talking about evolution, one of the strategies with sexual reproduction, the reason why you do that is to be able to adapt to a changing environment. And every method that comes up evolutionarily that provides more genetic variation, that provides more safety for that species to be able to adapt to a changing environment. And so if you go from, asexual reproduction where everything's a clone to sexual reproduction, then right off the bat, you already have two different sources of genetic information coming together. And then some of the offspring get, one chromosome from mom and dad. They might get, this one from mom, that one from dad. It's of the 23 or whatever, every one, it's a 50-50 chance that you get that information. But then if they also have the chance to cross over, then that's another layer of safety and ability to adapt, because you can also have different combinations of traits and things like that, because you can have those crossover events.
Right. Okay. Yeah. I'm used to thinking in viral terms because, they're like RNA viruses mutate rapidly. And so that introduces that change. Whereas in us, we have repair mechanisms. So if we mutate a section of DNA, assuming it's, not in a critical area, we can correct that and bring it back to what it should be. And so, the variation that may be introduced is, through crossover or through sexual reproduction of combining the mom's genes with the dad's genes, that kind of thing. And those lining up of, random. So, siblings aren't identical. They can get one copy from the dad, one copy from the mom. So that lines up all different. Yeah. That's really a fascinating thing to think about. And then to have like the sex chromosomes of pythons be so similar that they can cross over and exchange that genetic or the mutation and I think that's one of the coolest things about the morph thing is that you wouldn't be able to see that happen if you didn't have morphs. If we didn't have
same with parthenogenesis and androgenesis.
Right. Yeah. So we can see kind of a visual outcome that is not expected or not anticipated, that kind of helps us change our paradigm of what's going on in the genetics of pythons. Very cool. Very cool.
And what was the second term you said there after parthenogenesis?
Androgenesis. Yeah. So there's two different modes where an offspring can contain only genetic information from the sire. So even though this female laid this egg, this baby comes out. We had one this year that we did the genetic testing on, and then that individual actually went to Dr. Warren Booth. He's going to raise it up and see if he can get it to breed. But it got all its genetic information from the sire. So even though you saw that female lay that egg and it came out of that egg, it came from that female, it got zero of the genetics from that female.
That's insane.
So it doesn't happen very often. And the really cool thing in pythons and boas, what happens with parthenogenesis, so that's the female side where all the genetic information is coming from the female, that the only thing we've seen so far is an egg that only has one set of all of the chromosomes gets doubled. So if you had a het clown female that did parthenogenesis, then half the babies will be normals and half of them will be clowns. You won't get any hets. You're just getting a doubling.
Yeah.
But with androgenesis, there's two different ways it can happen. Both of them require the egg to be enucleated, which would be very weird, but it does happen. It could, expel both the polar bodies, or however you end up with an egg that has no nuclear DNA in it. A sperm comes and then doubles, then that's just like parthenogenesis. And you would get we call them like a half clone, or it's a doubling. You're not going to get a het for anything. And that's why a lot of partho clutches and andro is so rare. I would imagine it would be the same if we saw more of them, but lots of partho clutches, they're all going to be female. If we're talking about pythons and boas, if we're talking about colubrids and venomous, they're all going to be male because of the ZWXY difference.
Sure.
But so in ball pythons, we'll get all females and boas as well. That a lot of the time have health problems, and that's because they're homozygous for pretty much everything. So if there's anything bad there, it's
any deleterious genes. Yeah,
exactly.
Okay, that makes perfect sense.
So the cool thing with androgenesis, the other thing that can happen, I don't know if this has been shown specifically in snakes yet. I would have to ask Dr. Warren Booth. He's had multiple publications on this. But the other thing that can happen is two sperm can go in and fertilize that egg, and then you could end up getting that way you could make either males or females from androgenesis, whereas parthenogenesis you should always get females. So that's pretty interesting. And you could get animals if the female was a normal, or something that could not show up in the offspring at all. You could breed that male and end up getting offspring that, you don't even know that they're andro, because as far as partho goes, you would expect it to either be homozygous for it or completely negative. But with andro, you could get two different copies. And so it can be much more complicated, but it doesn't happen very often. So.
Right. But again, morphs kind of have lit the way for us to identify these things happen at all.
Yeah,
that's incredible. Yeah, I mean, I guess it's obvious to if you have a female that lays an egg and it hatches and she was never with a male, then parthenogenesis is occurring. But androgenesis, that would be, is it known in anything else, or?
I honestly don't know. It's such a rare thing. I when a breeder sent the shed to me for that and told me what the pairing was, and it was very clear it had all the genetics had to have come from the sire. Then I talked to Dr. Booth about it, and he knows way more about it than me. It's not something I've dug into. That's all that I learned from him. But I think that the way, at least humans and maybe mammals in general, some of the imprinting and the different things that we have that are important for our genetics and development, all that stuff, I don't think it's really possible for that to happen in a human, and maybe not in a mammal period, but I don't know. But if you start looking at fish and other stuff like that, it could be in some of those.
Very cool. Yeah. That's just crazy to think of,
Yeah.
It's fun stuff.
That's great. Yeah. So another kind of prompter for the show was, I was listening to a podcast that was new to me, right? It had been going for several years. And so this was an aged episode, and probably from 2.5-3 years ago. And they said, oh, Dr. Ben just figured out that desert ghost is actually these 3 different genes and the interactivity of these, blah, blah, blah. And so I heard sort of this explainer. I then asked you, and you're like, yeah, well, sort of. And so there's that specific question, but really as the question of, are you aware of genes that operate based on the interaction of multiple multiple defined genes? I know Nick Bunton has talked about his idea, at least, that the hypopresentation in Brettles is actually the sequencing of a couple different genes, something like that. So yeah, Thoughts on those ideas?
Yeah. So first of all, with the desert ghost thing, we thought at first that it was, first of all, we had a lot of pressure because that was like we had clown, we had pied. That was the next one. Like everybody wanted a test that would work for that. We had one that kind of worked like 85% of the time, but we weren't going to sell that because, it's not, but all the other tests we have are over 99%. But we found another marker that was on a different chromosome. And when we looked at the two different markers, then that increased our accuracy more to like 95%. And so we made Desert Ghost available as an elimination test. We called it something different. And we said, there's got to be at least one other mutation that's affecting this. And in the end, there was one other mutation, but it completely controls it. So those other two, the one that's on the same chromosome, it makes sense because of linkage that we're just seeing that because it's nearby. But the one that was on that other chromosome, maybe it helps with color or pattern or some other thing that we're selecting for as breeders who didn't even realize that we were, but it was somehow correlated with Desert Ghost. But once we ran, we got to that point and we're like, hey, we think this is polygenic and, blah, blah, blah. But then we offered to run 2 free tests for anyone that wanted to. So we ran, I think it was 552 or it was over 550 that we ran for free. And when we did that, we learned, okay, and that's when we found that third mutation. And then in almost all of those, we had a couple that were weird ones, but everything was all described by that one. So it did act like a simple recessive trait. Those other 2 didn't matter. But then to go to your other question, as far as defined traits that are part of like a polygenic trait, there was a publication, I think it came out this year, that was talking about, I think it was height in humans, and they had found over 10,000 genes that play a part in how tall you're going to be. So, that's buckets of data from a lot of different researchers over a lot of years to figure that out. It's definitely possible. And I think I remember like in school reading about ones that like are only affected by like 3, like something small enough that you could actually find all 3 and figure out, how much of the trait is predicted by that one part of the genotype or whatever. One that's kind of close to that is Labrador retrievers. So like black lab, yellow lab, and chocolate lab. That's 2 different genes on different chromosomes. One of them controls color, and then one controls dilution, I think is what they call it. And so it's really an interaction of 2 different genes in different places that gives you the yellow, the black, and the brown. So that's one that is technically polygenic because it's more than one, and it's a small enough number we can know exactly what those are and how to predict it. But in a reptile, I haven't seen that. I definitely like striping and like color in green trees and even in like I think almost the majority of the traits in like crested geckos and Amazon tree boas and stuff like that, they seem to all be polygenic or almost all be polygenic traits. So they're out there, but how many different genes control like blue versus yellow versus black in a green tree? I would love to have enough money to spend 5 or 10 years on that. That'd be fun.
What about like CRISPR and stuff to manipulate that and make an all black green tree python or something?
Yeah, that'd be very doable. What they did with the direwolf thing. What they're calling a direwolf. So to make that a white animal, they found a mutation, and actually the one that they think they found in the actual direwolf, they picked a different one because they didn't want to cause hearing problems or something. But yeah, we're getting to where we know enough about color and pattern stuff that we could. I haven't looked at like hypo or hyper melanistic stuff yet, but there are some that we know in homozygous state in ball pythons, like super black pastel and super cinnamon, stuff like that. We know what gene that's in, and it makes sense that it would add a bunch of that dark color and take away the pattern. And So yeah, we could do some designer stuff. That'd be kind of fun.
That'd be crazy. Yeah.
I guess, and this isn't meant as a trick. Why didn't you say that before? But it just sort of springs to mind. Whatever happened, so this is back in the time machine, whatever happened to being able to reproduce the look of the original platinum ball python, right? So it's always, the framing was lessers. Whatever was the deal with the sire to those animals that had a different look, was that ever reproduced? Was there a recessive trait that was in combination, Ralph's whole idea of like, oh, it's the normals that are the secret sauce, or whatever happened with that.
So yeah, he was right that the normals, so when he bred a platy daddy to a normal, half the babies were lessers and half of them looked like normals and none of them looked like the platy daddy. So that's, on its face, that's confusing. Like, why can't I reproduce this? And that's why he thought, there must be some recessive thing going on. Well, what's happened, and we don't have the sequence for this yet because it's hard to find the daddy side of that. There's lots of lessers, but to find platy daddies, people just haven't kept making them because it's kind of a pain. But what's actually happening is platy daddy, that one gene where lesser is, there must be another sequence that makes it the daddy part. So a platy daddy has two different blue-eyed Lucy complex mutations, but one of them does not make it a white snake. And in the heterozygous form, it just looks like a normal. And in combination with a lot of other things, it doesn't really do anything. But if you put it with lesser or butter or Mojave or something like that, you can make a Mojave daddy or a lesser daddy or a butter daddy. You can do all of those things. But yeah, so that's what happened was he bred platy daddy and then he got the lessers in that, but then he decided to breed the lessers to the normals in that. And when he did that, he would make platy daddies some of the time. And that's because some of those, about a fourth of those offspring would get the lesser mutation and the daddy mutation. And then you'd get a platy daddy. But yeah, people just haven't selected for that because it was kind of weird and people didn't understand it. And I still have people ask about it, and I have a few samples, probably like 5 to 8 that people have sent in that are daddy samples. But I just haven't taken the time to figure out exactly what that sequence is. But I definitely want to, just because it's kind of interesting and cool. But that's an example. Also, a lot of the time people think about, like, if it's that gene where Mojave and lesser and butter, all those are, then people think of that being a gene where everything's incomplete dominant, and that's not how genetics works. In that same gene, you can have something that's recessive too, or that is only going to show up if it's in combination with something else. And so that's the case there. If you have just a mutation for daddy and nothing else in the blue-eyed Lucy complex, it just looks completely like a normal. You can't tell the difference.
So you are finding all these hidden genes too.
Yes.
Hidden genes
that one is kind of a hidden one.
Yeah, that's crazy. So it just lines up, the right way to make that phenotype. And I mean, It seems like there's a few other genes that do that look kind of normal, but then they enhance, or they call them enhancer genes, or like diluter genes, or whatever they call them, and
how they interact with something, another mutation, right?
Yeah, like fire and vanilla, and there's a lot of them that, and even yellow belly will kind of enhance things in some combination. Yeah.
Cool. So I'm presuming the answer is no, maybe then out of this, just based on the complexity, the honestly terrible naming. Is there a double daddy? Has that been a homozygous daddy of that?
Super daddy?
Yeah, that's a good question. I don't know if he ever tried for that. I don't know if he had bred that platy daddy to any of his normals, then he would have had a chance to make that. That's a good question. I don't remember, because I did spend a lot of time on Ralph's page, because it was kind of during the same time I was doing my dissertation and doing all the stuff with TSK with their animals. So I went through and looked at a lot of his pairings. I don't remember him talking about that at all, whether he got like the super daddy part of that, or if he even had a chance at it. I know he bred the lessers to their siblings. I know he did that, and you wouldn't have a chance at that in that pairing. But yeah, that would be really interesting to breed a platy daddy to one of those normal offspring and see if you can get a super daddy. That'd be interesting.
Or even the two, the normal siblings, bringing the two siblings together.
The two daddies.
Yeah. that would be the way to get the super. Well, other than breeding into the platy daddy. Yeah. All these stupid names. It's hard to have a serious conversation talking platy daddy and queen bee and stuff.
Yeah.
Oh goodness,
it's crazy. Yeah. It's kind of humorous too. All the politics involved in naming some of these things too. But yeah.
Well, and that's part of the idea, right? And how it's super interesting that at least in some of these, they are genetically distinct, because that became a big part of the conversation was, is this just sort of labeling, is this a marketing gimmick, or are they actually different, and that was yet another sort of inherent bias to the analysis, right? Or the discussion.
Oh, yeah. Definitely. And Lesser and Butter ended up being one of those for a long time, until a month or two. I think it's probably about 2 months ago. We thought Lesser and Butter were the same. All the samples that came in, some were Lessers, some were Butters. They all tested the same. But we ended up having a few come through that were Blue-Eyed Lucies, but they did not test for anything on our Blue-Eyed Lucy complex except for Lesser. And so we sequenced and we found out in animals that were purchased directly from the Bells, they would test negative for the Lesser test. And we did actually find a Butter mutation. So it is actually different. And even though the Bells have sold tons of those through pet stores, as far as in breeders' collections, people, obviously we only have the sheds that people send for testing. Right. But breeders that are sending it in for testing, there's way more Lessers than there are Butters. But if you are going to people's houses and testing them, I bet that there's way more Butters than Lessers. So that's kind of a cool thing. And then in that same complex, you have the Phantom and the Mystic that a lot of people were confident was different, but they're genetically identical as far as we can see, unless there's a secondary mutation that travels with it most of the time or something weird like that. That definitely the causal mutation is exactly the same.
Yeah. Wow.
That's wild.
So now I remember the big, the big push, there's no such thing as snake genetics. It's just genetics, but sounds like there might be some snake genetics going on here where you can get recombination in the sex chromosomes. That's not human genetics, but I guess it's all genetic
for
principles.
Sex chromosomes. Yeah, that's definitely,
right.
You'd never see that in humans. They're extremely weird to see.
Right.
That.
Right. Huh. I don't know. That's kind of a old, one of those old things, there's no such thing as snake genetics, but
old sayings. Yeah. The cliches. So slightly changing gears, are there, do you have any thoughts on basically mutations that are taken to be inherited, a dominant mode of inheritance, but in reality, it's sort of actually there's a lethal homozygous form? In terms of what those could be.
Sorry. The spider, so the mutations in the spider complex, spider being one of them, Woma, champagne. There's quite a few of those that are like that, sometimes they'll be listed, spider could be listed as a dominant mutation. Really, it's an incomplete dominant. It's heterozygous for a leucistic. So when you get a super spider, it's all white. It's kind of like not zebras, jags in carpet pythons. So you can make a super, you can hatch it. Sometimes they'll even live for a day or two. I don't know of any. I think there was one that supposedly lived for 6 or 7 days, something like that. And it might be, is that kind of a similar thing as far as jags? You can maybe get one
chimera, right, that survived for some length of time, but presumably that was because at the fundamental point was not homozygous for the trait.
Yeah. So that definitely happens in ball pythons. I guess pinstripe we don't have a test for yet.
Well, yeah. So you're right. So you're right on it, right? Because Brian, I know it talked about. I forget if it was a male or a female, and he had come on it must have been on Reptile Radio, and he was he just kind of tossed it out there. And this is well after they'd been around. He just sort of had it oh, this is, you know, and I forget if it was male or female, but he said something like, I think there's, you know, that it's genuinely a dominant mutation because he had, you know, this is him talking, right? Quoting him. He said it was something like he had produced 16 or 20 offspring and all of them were pinstripes. And just the odds of that happening by random chance are so, you know, and everyone was sort of like, again, it falls into the, oh, this is a narrative. This is just Brian inventing stuff. He's just saying stuff, you know, no one cares about pinstripe anymore. He's got to make it interesting. But
so with leopard, we were able to prove that to be the case. So we do have a leopard test, and I've tested many super leopards. So, and as far as visually, you cannot tell the difference. People like Justin Kabelka, I asked him specifically once we had that, I was like, hey, you know, can you tell a difference? And, you know, even on the down low, I don't want to give your secrets away. He's like, no, I can't tell a difference between a leopard and a super leopard. So yeah, that's the best example I can think of that we know, like genetically. I've definitely tested. It was really funny. One of the very first few super leopards I got, I knew the breeder. And so I called him. I was like, hey, I had a couple of yours pop, you know, homozygous for leopard. Is that possible? Like from the pairing, was there leopard on both sides? He's like, funny you say that. That clutch is actually hatching right now. Let me slit those. So like while I was on the phone with him, he slit, you know, these 8 eggs. He's like, yep, leopard, 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 leopard. He's like, yep, it breeds super leopard also. So, wow. So yeah, that one we're very sure. Pinstripe, I've kind of heard that. Calico or sugar, you know, whether people just seem to never breed that on both sides. But I don't know personally. I have a few, but I don't have enough where I ever would have paired them together either. But either a super calico is lethal and they never, you know, early enough in development, we never see them. They abort or that egg crashes or whatever, or there is, but people just haven't figured that out, I guess. But yeah, pinstripe, I've heard that from at least 3 different people where they feel confident they have a super pin. So we need to get that test and then we can figure that out for sure. Stranger is another one that may be a legit dominant trait.
Okay. Wow. Okay, let's see. Going to page 2. Yeah, so then another sort of complex thing, and I guess here we finally get into suit Justin's fancy. We're going to fight a little bit, or at least get you going on some maybe some more ethically questionable kind of things that, you know, when we're, you know, listening. A lot of these podcasts, right, are often in the sort of leopard gecko context, ball python context, hognose now probably, you know, in terms of these communities that are heavily focused on mutations. And I know you've been on several of those shows and things. So the question becomes, getting into sort of the fact that deleterious traits, as we hit on way early, right, exist. In terms of the leopard gecko stuff, right, there's the enigma that there's the neuro functionality on it. It seems like it's probably the same or similar to the spider/jag idea. And then there was the lemon frost, right, that created this beautiful appearance, but then brings with it these, iridophore tumors, something like that. That
sounds right. Wild. Yeah. I've read about it. I haven't ever seen or even like watched a video about it, but I've read about, there's some publications about that lemon frost.
Yeah. Just sort of wild, right? That there are, you know, so often, and I don't remember if it was you or if it was Nick, you know, or if it was, Travis or who was talking about it, but there was just talking about sort of the rates of or likelihood of a trait having deleterious side effects, talking about the color mutations versus pattern mutations, and that generally speaking, it seems like color mutations are less likely to be problematic than pattern mutations, where you're getting disruptions that are going to disrupt sort of cell location or cellular functionality location.
Yes. Yeah. So I probably talked with Justin about this. This is when I was a grad student. There was a guy that did chicken genetics, and he overheard us talking about this kind of stuff, you know, spiders and jags, and what he described to me. And now I've read it in several papers, but that the first time I was exposed to it was as a grad student, and So he was describing that in very, very early embryonic development, the melanocytes are traveling with the neural crest cells. And so that's why a lot of the time a change in pattern also is a change in the neurological network. And so if you get this really whacked out pattern, you're also getting this really whacked out neurological, network set up.
Yeah.
And some of those are very bad for the health of the animal. Right. There's also with leucistic animals. So this, I guess you could kind of think of it as being both pattern somewhat and color. But yeah, with leucistic, whether it's mammals or reptiles, it's very common you have hearing problems and or eye. Like sometimes you'll get bug eyes or pin eyes. Those are pretty common problems across different, like in dogs, horses. Like it's not just reptiles. But yeah, I think that you're right. If you have something that's a color trait, it's less likely to cause problems than a pattern, because that pattern difference also, the cells that are traveling with it are the neural cells. And so that neurological network is jacked.
Yeah, they did publish recently on the inner ear issues associated with, was it jaguar? With the
I know there's a spider ball python.
Spider ball python. Yeah,
I know that there might be a jag one also.
You maybe assume it would be the same thing since it's kind of the same phenotype, maybe. But yeah, it's hard to say for sure.
Well, we know where to look, so we'll be looking in other python species. We'll do carpets as well. I don't know how fast it'll be, but yeah, we'll definitely be sequencing some jags and looking to see in that same gene if it's a similar mutation, and then we'll know for sure
on the homologous type chromosomes, that kind of thing.
Yeah, so albino is probably the best example. One of the common ones in humans is ACA2, and that's what lavender albino is in ball pythons. There's ACA2 mice. So yeah, like that's why that really early paper on albino, lavender albino, and ultramel, why they were able to find all three of those is because they went to the genes that they know caused albino in, cats and dogs and rats and humans and all that. And so they're able to find those mutations in those same genes also caused albino in ball pythons. So we do have a piebald test for blood pythons, and that's, in the same gene as or in blood pythons, it's the same gene as the one that ball pythons. It's a different mutation, a different part of it, but yeah. So I fully expect. Gene. Yes.
So you reduce the size of your haystack that you're looking for the needle in. Is that the deal?
Oh yeah. I think it'll be much faster to find. Like, I think if we go through and try to find albino, like in everything, and axanthic in everything, and I think that once we know where to look, and we just get a few animals of each species, and we can look in that one gene, then we should be able to find them a lot faster than when we didn't know anything.
And I mean, this might be a dumb question or just a clarification, but you can also see, like, if an animal's heterozygous, you can see the gene on one or the other allele.
Yeah. Usually it depends on how you're sequencing and how deep the sequencing is. Sometimes I'll have bald spots, like, whatever gene I'm really interested in. If the sequencing doesn't work really well in that area, I might have a bald spot, and I might either have no reads or I might only have a couple reads, and they're both normal looking, but it could still be het. I just don't have enough reads to. If I have over 20, then I feel pretty confident I should see that. But so yeah, it just kind of depends. But once we know where to look, then we can PCR just that one spot. Then we'll have a whole bunch of copies we can look at, and we can test and see if it's heterozygous or not. But yeah, that's part of the difficulty when you don't know for sure where you're looking, and you have to just sequence the whole genome. And to get that really good, you have to do 35 or 50x, and that's gets expensive to do very many animals that way.
Right.
Even though sequencing costs have come down, but once you have an idea where to look, then you can amplify just that small part. You go from 1.7 billion base pairs down to even 10,000. That's a massive that's a massive improvement in your odds.
For sure. So I'm curious too, just kind of mechanics of this. Like if you have 60 different tests, I mean, I'm sure you're getting, samples in. Do you kind of wait for a batch that's, large enough samples to kind of fill up a thermocycler or whatever to before you run the studies? Or, I mean, are you telling your customers like, okay, there's going to be like a week delay or do you get enough so quickly that it doesn't really delay much?
Thankfully, it wasn't like this at the beginning. At the beginning, it was what you just said. Like I would do a run like once a month or once every 6 weeks because everything was getting sequenced. And so the only way to do that cheap was to do big batches. Okay. But now we're able to, first of all, we have more volume. We have more tests available. Yeah. So we have more sheds coming in for testing than we did back then. But also we kind of have two pathways. So if somebody wants to have right now our big panel for morphs for ball pythons, you can get a panel done. You get 48 results. So we have 48 tests that are done. So that we still only do about once a month. So it's like 4 or 5 week turnaround for a panel, but you get 48 results. So if you don't have a clue, or if you think, hey, it might have some, mutations in here. And honestly, multiple times we've had people that paid big money and then found out the person they bought from wasn't being honest. And so they run panels to find out what all was lied about. So you get a whole bunch of data, but it does take longer because we have to batch them. But I think in the next year or two, we'll be able to cut that turnaround time in half as we, keep growing the volume coming through. We can do them faster. The other one, like if you just want 1 or 2 or maybe 4, even up to like 5 different tests, we can do that cheap enough where we do them most weeks. We'll do 4 days a week where we'll run everything that comes in. And so our turnaround for, if you're just getting, hey, is this a het clown, het pied, is it pastel? If there's 2 or 3 or 4 things you want to know, then we have a 1 to 2 business day turnaround. So it's really fast.
Wow. And is this like unique primer probe sets, or are you doing real time or QRT PCR, or what are you doing there?
Maybe.
Yeah. Okay. That's proprietary information kind of cool. I don't want you to.
It is not all the same.
Okay.
So some stuff we have to do a little differently because we're doing a lot of different types of mutations, but okay. At least some of what you're talking about.
Yeah. Okay. That was more of my curiosity rather than, I don't want you to say anything you don't want to say on here, but that's cool.
No worries. But it's all stuff that thankfully we can run like everything that comes in. Like our busiest day is usually Tuesdays because Monday they'll just, and this is all in Texas, but they'll prep everything that came in on Saturday and on Monday. They'll prep all of that on Mondays. And so we'll have really big runs on Tuesdays, and then, then we'll run every day the rest of the week, and we can just get through everything people sent in. And so it's usually, a lot of the time it's, 24 to 30 hours after it gets to us, we've already got results up. So.
Wow. That's really cool. You got it down to a science. That's awesome.
Got the science down to a science. One thing that jumped out right when you mentioned the context of that the gene location for piebald and blood pythons is the same as ball pythons, but the actual sequencing is different, right? Not that it was a concern in this context, but that's an illustration or proof, right, that it's not a hybrid from a ball python. It's not that the trait was, brought over from a ball python because the sequence, the mechanism is different, right? So that was, when you said that, it's like, well, that's really cool, to anyone out there, casting doubt or aspersions or whatever, obviously from nothing, but it's not.
That one is the only one I can say right now. I know, but as we do more like pied and berms and pied and retics, like we'll be able to tell if it's the exact same mutation as one of the others.
We can't necessarily be evidence that it is, right? That it could go either way. But if it's different, then it's sort of exclusionary evidence, right? Of saying, okay, well, it's not just the same.
But if it is identical, then
pretty skeptical.
Yeah, very skeptical.
Wouldn't you be able to see that, like, there's, a mixing of berm and, the
that's
other
what I was
genes?
Say.
Okay.
That would make me skeptical enough that I would be willing to sequence. I would sequence whole genomes of a few and be like, oh, okay, we see this too.
Yeah.
But yeah, the chances of, like, in that whole gene, some genes are, 60, 70,000 base pairs long or whatever. But to have the exact same mutation pop up 2 different times is just mind-boggling that would actually happen. It's much more likely that somebody crossbred 2 species and got that mutation in.
Right.
Very cool.
So if you bred a piebald blood to a piebald ball python, you'd have double hets because it's in different areas?
I would imagine they would still, I would imagine they'd still look like a piebald. But I mean, it technically it is, it could be affecting a different domain. And so maybe it could, the one normal copy in one spot could make up for the mutation, and the normal copy in the other spot could make up for the other one. But I think it would be more likely, like with Ultramel, we have 2 different mutations in 2 different spots. But if you have a double heterozygote, it looks like if you're homozygous for one, homozygous for the other, or have one copy of each, they all look the same to the breeder. We can't tell the difference. So I would imagine that if you bred the pied blood to the pied ball python, you'd get all pieds, but that would be kind of fun to try.
Yeah, that's interesting.
So another thing that you had mentioned previously when you were talking about the context of the panels, right? Running everything that's in there. So it makes me think of, and I'll put it kind of a semi-pin in this. I have a discrete question and then someplace to go. When you're talking about misrepresentations being evidenced by those panels, are those misrepresentations of, like, inclusion or of omission? Right, because I know in the leopard gecko context, sort of the new wave of, and it's probably true in corn snipes, or at least it's going that way. Maybe it's not as the sentimentality of leopard gecko keepers is not quite, of corn snake keepers is not the same as leopard gecko keepers, but there's a desire to have something, no unknown hets or no known hets, and these sorts that just only like this strain
only of albino for one type of albino.
Exactly right. And that you're, meanwhile, the folks who have validated that information have produced a ton of it, flooding the market, or flooding into the universe all these animals that are deliberately both a mixture of both things that then get pushed out into the community. The ethical challenge associated with test breeding compared to your tests and things. But I was just curious, is that in the ball python thing, were you thinking of, it's a possible het X and it's not, or is it, no, there's no possibility that albinos had albinos in the mix? Which of those is it more frequently?
It's way more frequent in ball pythons that they didn't get everything they paid for. And it happens sometimes. It happened to Justin and I that they get something different than what they paid for. So Justin and Justin has an even longer track record of bad luck with albino stuff. But so we bought a pair of het albinos, and Justin, called. I think you called me first. Eventually, sent me a picture. I'm like, what is this thing? Like, it was so weird looking because we were expecting an albino. Ended up being a hypo, and I guess that guy said that, I guess I mixed them up. I gave you the hypos instead of the albinos. I don't know what happened, but there's definitely mix-ups that happen. But the one where the big problem is people buying these animals that are multiple incomplete dominant things, and then they're supposed to be triple het for, pied, clown, and desert ghost or whatever. But they were either not het for anything or only have one or two out of all the ones that they paid for. So that person, I think the last time I checked, is still selling animals, and I don't know.
Oh my goodness. I guess that brings up the other question is, should you be ratting this guy out, making people aware?
I definitely won't. I tell people that I treat it like I'm, their doctor, for their human health. I'm not going to give away.
Not going to out their spouse when they give them herpes or whatever. Yeah. This guy's cheating on his wife. Yeah.
if they prefer me to be the one to talk to the breeder, like, sometimes I'll know the breeder better than they do or whatever, I'm happy to do that for them, to, be part of that conversation or confrontation or whatever. But yeah, I would never, just like start talking to
take that
someone
upon
else.
yourself.
Yeah, I'm not going to do that.
You don't need that headache.
No.
Right. It's a funny industry in some ways, especially when you get into stuff like that. It's like I've seen a few posts recently where people had paid for snakes and hadn't gotten, and now they're airing and saying, "Hey, this this is a problem." And then all of a sudden, all these other people are like, "Oh yeah, he did that to me too." And it's like, if only there was a way to prevent that earlier on, But
so
I guess that's
people.
Yeah.
Oh yeah. People that are buying these, $5,000-$10,000 animals now, ball pythons, even if they know what it is. There's one guy in Vegas that's bought a whole bunch of stuff, and he's so funny. He just loves even though he knows what it is, he wants to have it tested and make sure it's what it should be. But yeah, like even if you're pretty confident in what the breeder says, still sometimes weird things happen. Like Partho, we had one, as actually someone I know really well, a friend of mine. He bought a leopard het desert ghost, and it came from a pairing. The sire was a desert ghost, a visual homozygous desert ghost. But he had me run stuff for it for other things, and it just came up like completely negative for desert ghost. And in the end, what we found out, actually this is another time with the super leopard thing. The female was a leopard, and when we ran this animal, it was a super leopard, and it was negative for desert ghost, and the female didn't have desert ghost in it at all. And so that's when we figured out, you got Parthoed, bro. So there's no desert ghost there, even though it has to be, because the sire was the only sire in there. It has to be desert ghost. Well, it's not desert ghost. It's a Partho. So we did a whole genome on it and showed, the heterozygosity is crap. You definitely got a Partho. So.
Wow. Well, I guess you got to keep that in mind when you're, because you could go and slander this guy, and he's like, "No, I paired these two together." You got to be careful, I guess.
It's an honest mistake,
right?
And he was really mad, and talking about how bad genetic testing is and all this stuff. I'm like, I think in the end he believed us, and I wrote him a nice thing. I was like, "Hey, if you see this in this clutch, if you see this, like these are the things that you need to worry may not have desert ghost in them. If you see this, then like if it's a male, it's good.
Right.
If this or that, then you're good. But these are the things, if they're not there, or if you have those, then they might not have desert ghost in them." And
right. Yeah, I guess that could be, for both the buyer and the seller. Like they could both be mad at you. Like you're giving them these results, and you're screwing my business, buddy. And I know what I paired, and
And we've had several. Sometimes people send their axanthics, because we can test for 3 different lines of axanthic in ball pythons, and so they'll send them in. And they think that it's one, or like it's completely negative. Like, well, I bred them. It's an axanthic male that I bred to it. But then we run the other 2. Like, oh, it's not VPI, it's TSK or whatever. So that's happened, and so fun stuff.
Keeps you on your toes, I'm sure. Hopefully that makes it easier for the stuff that happens down the road. Oh, this happened before. I know how to answer this, or I know what to test for to, verify what's going on. Tricky
for
stuff.
Sure.
In terms of designing tests for the corn snake mutations, how are you kind of coming to pick which mutations you're doing? And is that do you see any market for that moving in the vein of sort of the leopard gecko, no unknown het sort of ideal, where they just want it to be sort of only this thing and not just, the random pet store corn snake could run the panel on that. It could be anything, right? Almost. So.
Yeah, for sure. Yeah. I definitely have heard breeders talk about that more in corn snakes. They're more, they would rather it be just this and not that kind of a thing. Ball python breeders in general just want everything they can so far. I'm sure at some point that will change, but except
the
for
market
pastel,
matures to a point where, it's like...
Yeah. With pastel, sometimes people do test because they want to make sure they have a pastel, not a super pastel, So if it's a super pastel, they know everything they make is going to have pastel. And so sometimes they'll choose that so they can, not get the super. But yeah, I think, and as far as corn snakes, like the cool thing with them is there's been some really good work where they've also CRISPR'd some of these mutations. Scaleless and striped, and I think maybe one other one. But not only did these researchers find the mutation that causes it, but they also went in and CRISPR'd them and generated animals that had the same phenotype. So that was pretty cool.
Wow.
They've done some really cool work in corn snakes.
Yeah.
So that's helped. But yeah, as far as choosing, really what we do is we talk to breeders and say, hey, what's going to be most helpful? We look at Morph Market and see, if there's a truckload of this or that on there, like in ball pythons, pastel, whatever, it's all around. People aren't maybe necessarily excited about a pastel test. You can tell it a lot of the time, but there's so many pastels out there. We do actually have a lot of people that want to test for pastel. So, we kind of look and see what's on Morph Market and then ask breeders what would help you the most. And then that's kind of, we usually have like a top 10 list that we're working through. So.
Very cool. Yeah.
That's incredible. I was curious too. This has been a while back when they discovered or sequenced or identified the virus that causes IBD. And at the time, like only a couple reptiles had been sequenced, species. I think boa constrictors had recently been sequenced, and that's what allowed them to find that viral sequence in among the boa sequence. They could rule out what was boa constrictor and what wasn't, kind of thing. So when they had the full boa sequence, is that still a problem, or have lots of different species been sequenced to the full, genome or large part of the genome at this point?
I think there are. I know snakes better than like reptiles in general.
Sure. Yeah.
Snakes, I think there's a chromosome level Thamnophis and Elathi. So I think it's corn snake and garter snake that there's chromosome level. But some of the bioinformatics people we've been talking to said that it's still, there's a big difference between whole genomes that are built only with short read or Illumina stuff versus ones where you do some short read and some long read.
Okay.
So the bioinformatics people don't like the ones that are generated with only short read. I think that there are very few reptile genomes that have long read also.
Okay.
There can be some chromosome level ones that are okay. But like a really good one, like we spent, I think for us back when we did it, we probably spent between $40,000 and $50,000 doing a whole male ball python genome and a whole female ball python genome. Very, very deep and with lots of long read to help make sure everything got,
put in the right order.
In the right order. Yeah. So it's really expensive and it's hard to get the money to pay for that in reptiles because we're not feeding people with reptiles.
Yep. Yeah. That is a challenging thing. I'm just so impressed with how much you guys have done and what you've come up with. It's really just impressive. Really cool. Yeah. Congratulations.
It's been a fun ride.
Yeah. Right. Nice.
But yeah, sequencing is getting cheaper over time. So maybe another 5 or 10 years, it'll be even easier. I hope that'll be the case.
Yeah.
But it's definitely slowly, like 10 years ago versus 5 years ago versus now, we're getting more happening in reptiles, but it is still difficult to have a really high quality, deep, long read whole genome.
Yeah, I think I'm really impressed with the fact that they can, CRISPR a corn snake and, they've developed those techniques to, however they're doing that. I imagine it's got to be at the single cell stage, or the egg and sperm stage, or can they modify once it's developed a little bit?
So the way they are doing that, I actually was reading that here fairly recently. They're doing a surgery, and they're going in the egg stage. So they're going into follicles that are I believe they're pre-vitellogenic, so they're not vitellogenic yet.
Okay.
So this is like at a stage, like if you're ultrasounding, they probably still look like a water balloon. They're not starting to go white yet.
Yeah.
But what they're doing is doing a surgery, cutting open, going in and injecting the egg. So in that case, you're only going to potentially make a het. And then what they were doing is they would go in, since this is a mutation that they already, have a bunch of scaleless or stripes, corn snakes. So they could go in, take a normal female that they know is not het for stripe. They would cut her open, inject, and then breed her to a stripe. And then they could see how many of the babies were stripes.
Right.
And they know that had to have come. And then obviously they can do the sequencing to, triple check to make sure it's their edit that got in. But
wow, that's cool.
But the other really cool thing they've done, I don't know if you guys saw this, Beth Roberts at the Memphis Zoo. Did you see their AI? They're able to get a sperm sample and store it, and then do AI, and they're working with Louisiana pine snakes.
Oh, cool.
So that's pretty cool. Yeah.
Yeah.
Really like seeing that.
I'd seen some AI had been done, in the past for different species, but it seems pretty rare and difficult. It's not, I guess, anytime you take a new species. I know, like a researcher at Utah State, Dr. Wang, he's working with hamsters, and apparently CRISPR on hamsters is not like mice. It's very difficult and very challenging, and all the different things he has to do to get that to work. And he's one of the only people in the world that has gotten it to work and gotten transgenic hamsters through CRISPR-Cas9. So, you would think, oh, a hamster is just a big mouse, no problem. But yeah, it's completely different. So yeah, it's cool to see these things work and people developing different strategies, especially in regards to, endangered species or, extinct species. The idea of bringing something back if it's gone extinct is kind of cool.
Yeah, fun stuff for sure.
Yeah. Well, it's getting late for you. I don't know if Rob has any more burning questions or if.
Yeah, we're almost. So my last question, and we've just been in this same sort of circle, is: do you have any personal thoughts on like the green fluorescent protein put into axolotls or the glowfish? These just in terms of as animals that are now out in the hobby, you go to PetSmart, right, and they have these glow danios, and there's pink ones and orange ones and green ones, and as I say, the glowing green axolotls that are they're cool, man. As much as it's sort of antithetical to a lot of my fundamental beliefs, they're pretty cool.
Yeah. I mean, as a someone that's done a lot of gene editing, I think it's cool and interesting, and I can see why a lot of people like it. I've seen, even a natural thing like having a scorpion and just turn a black light on. Like if you're, at someone's house at a party and people are hanging out and you do that, everyone's like, oh, like if you can have it be, some of them be pink and some of them be green, some of them be red, like that's even better, Right. But yeah, I mean, the studies, like we had to do some FDA stuff for genes we're putting in, and some of them were GFP and stuff like that. And, all the safety stuff, it seems like it's not, it seems pretty dang safe and to not cause a problem for the animal. But I've never done the studies myself or looked at it myself. You never know if they're, saying that because they want to make them. But
right.
I think that there's enough done by enough different people that it's a very safe thing. So I don't think it is really affecting the well-being of the animal, except for maybe the type of person that would buy it is maybe going to handle it differently than if it wasn't a glow snake or a glow fish or a glow whatever. That's the reason why I would be a little hesitant. I think I would be happy to help somebody else do it, but I wouldn't want to be the face of that.
Right. Well, well, it seems like like a GFP, it was more of a research tool, like kind of like using a morph as a marker of how different traits or different modes of inheritance or whatever, you're using that as a marker for another question that's more genetic or at the genetic level. So,
that's why
I think
we used it was, we would have GFP linked to whatever protein was important for us.
Right.
And we saw some GFP, we knew our other one was there too, it wasn't, and it supposedly isn't causing any problems, but it makes it easier for us to select and move forward.
Exactly. Yeah. And so we're just lucky that we have all these crazy mutations popping up in nature, and that we can find all these cool things about what's going on in our reptiles. So I don't know, it's pretty amazing. And, adding those, experimentally is maybe, similar to finding a morph in the wild to some extent. I guess they're not generating jellyfish glow goo, whatever you want to call it, luciferase in their genome. But I guess it's was it you that would usually say, it's snakes in boxes. We're not rewilding we're not doing anything that's conservation. So it's not
I remember having that talk with Nick many times.
Yeah. I still get a kick out of the thought of, once you remove it from nature, is it even the same species? Because you've removed its evolutionary trajectory and you're selecting who it breeds with. So it's no longer a part of that, and it's drifting, it's moving away from the species. So can you even call it that species? I think that's such a fun, experimental thought process to
me. Yeah. That's the biggest difference to me, like personally. And I can understand people having other opinions and the conversation, and I like being a part of it. But like, to me personally, like, as soon as you're making a breeding decision for an animal, that's not a natural species anymore. To me, that's not pure. That's not wild. That's not like if you say something's a locality, I can still feel comfortable with that. Sure. But like,
saying it's a pure species.
Yeah. I mean, a human made that breeding decision,
Right. Yeah.
It doesn't seem wild to me anymore. It doesn't seem pure to me anymore.
And you're not letting predators pick off the weak genetic or the ones that stand out, or, the ones that blend in or not surviving longer to produce more that fit better in the environment. So,
and spoiler alert, guess what happens when you preserve the parts of the species that don't have strong traits? You get a decrease in reproductive rates. You have fertility problems.
Right.
What's happening to humans?
Right.
Kind of a thing. But I would do the same thing. If it's my kid, I'm going to do everything I can to keep them alive. So.
Right. Yeah. Oh, that's interesting. I thought about too, asking you about inbreeding and things like that, but we'll save that for another day, because, that idea of how genetics play a role in that, if you have strong and balanced genetics and there's no deleterious things, then inbreeding's not such a big deal, but
if you're optimized for the environment you're in, then asexual reproduction is great.
Yeah. Right.
If things change, that's when, if you have a long generation interval and things change, that's a bad mix.
Yeah, no, that's true. And then they disappear. I mean, I guess we just went and saw the new Jurassic Park. Regardless of how crappy the acting or the story or the plot is or the plot holes, it's still just freaking cool to see dinosaurs on the big screen. I just have to see those movies, in the theaters. But, thinking about all the species that have lived on Earth, most of them are extinct. They're not still here. We have a fossil record of some. And that was one thing I was going to kind of mention. I ran across this study where they found these little reptile species it was a pretty full fossil, and it's like a 4-inch little lizard, but it has these huge, like, fins or appendages off of its back. What was it called? It's and I think Miss Mirasara Gravogeli. So it's like a Triassic little reptile, but kind of interesting. They just discovered a full thing and showed that it's these scale, or hyoid appendages that's come out of the back. Kind of interesting things. So, yeah, I may have some of those terms wrong, but yeah, it's paleontology is not my strong suit necessarily, but yeah, it's really cool to see that, thinking about all the diversity of reptiles that we missed out on. I guess that fits in with the topic a little bit, but yeah, cool stuff.
That's cool stuff. Yeah. And I'm the same way. I haven't seen that newest Jurassic World now, but yeah, I have to watch them. That whenever people ask me like what my favorite movie is, or especially if they say like most impactful, definitely Jurassic Park.
Right.
There's nothing that was emotionally impactful like that scene when they see him the first time, when they're in the Jeep. Man, there's no movie that's ever affected me like that part. It's like, man, that would be so awesome.
Yeah, for sure. Yeah. Well, I wonder, down the road if that'll be a possibility, if we'll be able to resurrect a species like they did in that movie. It's probably not the same background or ideas. But.
Right now, the people that do that, they're saying that, once you're past, I think like 1 million years, like being able to, the DNA is so fragmented, no matter how it's been preserved or whatever.
Yeah.
You just can't get enough to be able to piece it together. But like we said earlier, that's what science is, as we figure out that what we thought before was wrong. So
right. Yeah. I mean, the dire wolf kind of misrepresented itself a little bit, where we have dire wolves. Well, kind of. You got a percentage of a dire wolf. Yeah. So
that's a fun conversation too. But interesting. Yeah. And it's cool. Some of the stuff they did is definitely groundbreaking. But calling it straight a dire wolf with a straight face is weird to me.
Yeah. Right. Well, thanks so much for coming on, Ben. It's really awesome to have such an expert in genetics and being able to pick your brain for a couple hours here. And just really appreciate your time and effort to help us understand what's going on with our snakes in boxes. We appreciate it. Yeah.
Well, it's been fun being on, and our pre-conversation. Definitely, I hope in the next year or two I'll be able to go on some of the field herping trips. I definitely miss doing that. That's been something that I haven't done very much of that I would love to do again. So
some great memories herping with you. So got to make that happen again sometime soon before we get too old to lift logs and stuff. Where can people find your information? If they're living under a rock and don't know about RGI at this point, where can they find you?
The best place to see what we're doing and contact us would be through Instagram or Facebook. We do have a website. You can. Previously, we only had our tests available through Morph Market and Clutch, but now you can actually buy tests directly from our raregenetics.com website.
Cool.
And then we do have every Tuesday night at 7 p.m. Eastern, we have a YouTube show, Reptile Genetics Weekly, that goes up. So those are the best places to see us, what's going on. And as far as getting in touch with us, just messaging on Facebook or Instagram is a really good way to get us.
Nice. Yeah, I've watched a handful of your shows and just go over cool topics and talk about what's going on in the world of reptile genetics. It's really cool stuff, so check it out. All right, well, again, thanks for coming on, and we'll thank Eric and Owen and the Morelia Python radio team, and we'll see you next week.